in the processing of functional and nutraceutical cereal and extruded products. I will introduce our team members, which is me, Aina Shahira, Rael, and Hanif. Okay. For the introduction, cereals such as wheat, rice, corn, barley, and oats play a central role in feeding the world population, since life endosperm of cereals is mainly comprised of starch, making them humanity's primary nutritional energy sources. Nearly a century ago, grain foods were first introduced by the first USDA food guide in 1916 that promotes starchy foods as part of healthy eating plan. The germ of most cereal grains is rich in oil and lipid-soluble vitamins, while the outer layers have high
Sorry, please make the slide in slideshow, yeah, mode.
The germ of most cereal grains is rich in oil and lipid-soluble vitamins, while the outer layers have a high content of cellulose, hemicellulose, and minerals. Furthermore, cereals are also considered as good sources of many bioactive components such as dietary fibers, which is lignin, inulin, and resistant starch, phenolic compounds such as lutein and xanthin, anthocyanins, vitamins, and minerals. In addition, the byproducts of food industry are also recognized for their nutritional value due to these health beneficial constituents. Since these byproducts can be utilized as functional ingredients in food product development, the recovery of food waste is expanding around the world in order to alter the characteristic of cereals and transfer them into ready-to-eat foods. A series of traditional technologies is applied such as dry milling, extrusion, fermentation, use of enzyme. Moreover, the new technologies such as encapsulation are being explored in cereal food processing to enhance their nutritive value. Next slide. For the cereals and their food application, I will explain each of them. But for in general, all cereal grains have similar parts which are present in approximately the same proportion. However, the components and structure of these parts vary among different species that result in distinct oral, sensory, and nutritional values. In the United States, recently the use of corn are used for non-food applications such as bioethanol, sugar production, and ingredient for paper and pharmaceutical industries. For the wheat is one of the first domesticated food crops and occupying one-third of the total cereal sowing area in Asia, North America, and Eastern Europe. Modern wheat cultivars usually refer to two species, which are hexaploid bread wheat and tetraploid hard or drum type of wheat that used for macaroni and low-rising bread. Wheat has high starch content, which for whole grain, the starch content is 60% until 70%, and for white flour, the starch content is 65 to 75%. The protein of content in wheat is usually 8 to 15%, with some essential amino acid being present in higher amount than what is required nutritionally, while lysine is deficient. However, wheat products have been blamed for a number of adverse reactions for humans, such as intolerance and allergies. Next, this rice is also type of grain. Rice is the main staple food for half of the world population. 90% of the total rice sowing areas in Asia, since rice has higher heat and water growing requirement than wheat. Starch accounts for approximately 90% of total dry matter in rice seed, followed by a fraction of storage proteins. All the essential micronutrients are almost stored in the husk, aileron, and embryo of rice. However, there is reason that makes rice unsuitable for consumption, which is the micronutrients are removed during the polishing process since the oil-rich aileron layers turn rancid open storage. Next slide. Next, for the corn. Corn is popular in Africa and Central and South America since the role of corn as a foodstuff is important there. Corn kernels can be consumed of the cob, parched, boiled, fried, roasted, ground, and fermented for use in breads, porridge, gruel cakes, and alcoholic beverage. Corn contains about 72% of starch, 10% of proteins, and 4% of lipids. The typical yellow corn has many important vitamins such as vitamin A and E, while the minerals found are phosphorus, potassium, and magnesium. Even though corn kernels supply many macro and micronutrients for the needs of humans, some of the essential nutrients are poorly balanced that make it unsuitable as a major food. For example, it leads to the lack of essential amino acid, lysine, and tryptophan and ascorbic acid. For the barley, barley is one of the type of grain. Due to its superior heat and drought tolerance, barley is known as the fourth most widely cultivated cereal. For food uses, barley grain is abbreviated to produce pot or pearl barley. Whole barley grain consists about 65 to 68% of starch, 10 to 17% of protein, 4 to 9% of glucan, 2 to 3% free lipids, and 1.5 to 2.5% of minerals. Among other cereals, barley has the lowest levels of starch and fat. Barley glucan exhibit health benefit by reduction of blood cholesterol and glucose, increase weight loss by improving satiety, and indirectly control heart disease and type 2 diabetes. Next, oats. Oats are mostly grown in Europe and North America. Oats are distinct among cereals due to their multifunctional characteristic and nutritional profile. Furthermore, oats provide a good sources of dietary fiber and has been proven to be useful in the treatment of diabetes and cardiovascular disorder. Adding oats to normal diet of patient that has hypertension can reduce both systolic blood pressure and diastolic blood pressure. Oats have higher lipid content and oat lipids have a high proportion of oleic acid, which is preferable for human nutrition. Next. Last but not least, cereal byproducts is also the type of grain. In cereal production, hard bran and germ are removed during milling and processing to cater consumer taste. Wheat bran it is a concentrated sources of insoluble fiber, minerals, vitamin B6, thiamine, folate, vitamin E, and antioxidants such as phenolic compounds. It is an important factor for dietary element that reported for the benefits of cardiovascular disease and colon cancer prevention. Rice bran, phytonutrients have shown promising disease preventing and health related benefits. For the oat bran, good sources of vitamin B, protein, fats, minerals, as well as heart healthy soluble fiber glucan. In this slide, there are three byproducts, which are wheat bran. Uh, uh, can you please to the previous slide? For the cereal byproducts, there are three, which are wheat bran, rice bran, and oat bran. Next. Next, for the novel technologies in the processing of cereal-based products. For the dry milling, the cereal dry milling industrial unit composed of 4 stages, which are reception and cleaning of grains, conditioning, size reduction, and classification and separation of fraction. The grinding part is the central point of milling, in which the roller mill is the most popular. By applying the dry milling, the size of cereal can be reduced, and the starchy endosperm can be separated mechanically from kernels in the traditional process. Dry fractionation is energy efficient that require no solvent removal and recovery, thus require lower capital investment. There are several methods of dry fractionation of cereal have been developed, such as purlin, roller milling, and milling followed by air classification. Purlin is the process of removing the outer layer of cereal grains and typically used as an abrasive dehulling device such as Barley peeler that can provide gentle surface abrasion to grains with minimum breakage of residual kernels. Past research pointed out that dry fractionation could allow for full-scale separation of allulose cells from wheat bran, yielding a fiber-rich concentrate that potentially contains whole grain kernel bioactive compounds. Roller milling employs a special mill that allows grains to pass between rotating rollers for grinding and flaking, and went through sifter for fraction separation. Roller milling of hulless barley was applied to generate fraction with highly variable glucan and arabinoxylane content. To produce high B-glucan cultivars, yields more than 20% for whole barley basis of a fiber rich fraction with big glucan content of more than 15%. It can be obtained with a simple short mill flow. Air classification separate flour based on difference in density, mass, and projected area in the direction of flow. Moreover, research has proved that dry milling can be used as a potential effective method to reduce mycotoxin in cereal product. Reduction of mycotoxin in the milling fraction of corn flour can be done by removing of cereal germ, hull bran, tip cap, and some endosperm. Next, I will pass to Hanif.
Okay, the next is about extrusion. Extrusion refers to a process by which a liquid or semi-liquid product is forced through a die opening of the desired cross-section. Food extrusion has been divided into the general categories. The first is forming and the second is cooking. Now, for form application, low shear extruder are used to mix and form the desired product shape with minimum energy input. The product include pasta, cold form snack, and other unexpanded pre-cooked pellets. The screw typically has a deep channel, 20 until 30% of diameter with no compression ratio, and operates at a relatively low speed, below 50 rpm. Cooking extrusion normally utilize medium shear and high shear extruders, and significant energy is provided to the product through viscous heat dissipation, heat transfer through the barrel, and sometimes with steam injection. The screws run at relatively higher speed, uh, with a shallower channel depth. Next is enzymatic treatment. Enzymatic treatment, owing to their highly selective function and lower risk of undesirable site reaction, have been widely used to improve nutritive value and functional properties of cereal components and products. Also, the enzymatic modification of cereal protein starch and phytochemical have been extensively reviewed. The aim of this section is to scrutinize the recent development in the application of enzyme in cereal processing, and to summarize the knowledge generated from this research. Next is protein modification. Storage protein, including prolamine and glutenin, are the major proteins in general in cereal grains such as wheat, corn, and barley. Due to the large proportion of hydrophobic amino acid, cereal storage protein are insoluble in aqueous solution and easily form aggregates that interfere with functional properties. For the food industry, cereal storage proteins are normally produced as the byproduct of food processing, such as starch extraction and barley brewing. Utilization of this inexpensive byproduct in food formulation requires modification technique to improve their functional characteristic. Enzymatic modification of cereal protein has been the focus of a large amount of recent research. Enzymatic hydrolysis and deamidation are the most widely used enzyme-based method to improve cereal protein functionality and bioactivity. Therefore, two methods will be addressed in this section. The next is protein hydrolysis. Three main changes occur during the enzymatic hydrolysis of food protein. The first is peptide bonds are broken down and lowering the molecular weight of proteins. The second is protein surface properties are influenced by greater exposure of the naturally hidden hydrophobic residues. And the third is some microchemical changes occur such as deamidation of glutamine and asparagine residues that add extra charge to the protein. Next is carbohydrate modification. Starch and dietary fiber are the major component of carbohydrate in cereal grains. Starch is the main storage carbohydrate, whereas dietary fiber are the structural component of cereal grain. Resistant starch is the health promoting fraction of starch with low glycemic index, which performs similar psychological function as dietary fibers. High amylose starch, particularly from corn, is the most important source of the native resistant starch. Although resistant starch can be produced by physical method, the enzymatic approach is more common, in which starch is first degassed with alpha amylase, glutenized, and then debranched using pullulase or isoamylase. The next, Rael.
Okay, I will continue my fellowship friends presentation. So the next is about fermentation. Next. So fermentation is one of the oldest processes used by humans to improve shelf life and sensorial quality of cereal products. The preservative role of fermentation depends on chemical conversion of cereal components, specifically carbohydrates, to alcohol, acid, and carbon dioxide. Fermentative activities of microorganisms have been classified based on the end products into four main categories. The first is alcoholic and then lactic acid, acetic acid, and last is alkali fermentation. In the recent advance in microbiology and food processing have provided the opportunity to utilize fermentative microorganisms as starter cultures and the ability to trigger and carefully control the fermentation process, thus eventually improving the nutritional and health enhancing properties of the final product. So in the fermentation, there are probiotics and prebiotics. So the probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. So these microorganisms improve the balance of intestinal microbiota by changing the intestinal pH and generating antimicrobial compounds such as bacteriocins, organic acids, and hydrogen peroxide. The main bacterial cultures presented as probiotics are the Lactobacilli, Bifidobacteria, and Eubacteria genera. So probiotics is the positive contributions of this bacteria in intestinal health is highly affected by the presence of probiotics. And the probiotics are a wide class of oligosaccharides that are indigestible but fermentable but fermentable by intestinal microflora. So the effect of fermentation on nutritional quality. The fermentation improves but the level of phenolic compound bioaccessibility and their antioxidant capacity. The ferrolic acid, the major phenolic compound in wheat grain, showed significantly greater bioaccessibility in an in vitro model of the human colon following a combinatory bioprocessing involving yeast fermentation and cell wall hydrolyzing by enzyme treatment. The effect of fermentation on folate has been the subject of several studies, and the results have shown enhancement of folate delivery to blood circulation as a result of yeast fermentation of cereals. So the next is about the thiamine and riboflavin content reportedly decreased during baking, but yeast fermentation prior to baking elevated the total retention of thiamine in fermented bread. However, losses have been reported for vitamin E, tocopherol, and tocotrienol content due to oxidation during sort-of preparation and bread baking. Bread. So the next is about fermentation in gluten-free cereal products. In celiac disease, the inflammatory immune response is triggered upon ingestion of prolamine proteins from wheat, barley, and rye. During digestion, a family of peptides rich in pro uh, and glen are released. These peptides bind to the antigen presenting cells, which results in antibody production. In the last decades, various gluten-free formulations with more focus on bread dough have been evaluated, and several methods were proposed to overcome some technological and nutritional issues. Some formulations, for example, are mainly based on starch, so these products suffer from fast selling and low nutritional value. The modification of protein digestion through proteolysis seems to be the most effective method to reduce the allergenicity of the gluten. However, this would compromise the viscoelastic properties of the protein network, and the approach is not well demonstrated. The selected strains of lactic acid bacteria reportedly reduced the allergenicity of gluten in rye flour. So the next is about microparticles and nanoparticles based on cereal proteins for nutraceutical delivery. So in the last few years, encapsulation techniques using cereal proteins have been developed as nutraceutical delivery systems to create novel functional foods. Aside from being a vital macronutrient in foods, proteins possess unique functional properties, including their ability to form gels, films, and emulsions, offering the possibility of developing delivery systems for both hydrophilic and lipophilic bioactive compounds. In the past decades, zen and gliadin have been prepared into microparticles and nanoparticles, incorporating unsaturated fatty acids, vitamins, minerals, essential oils, antioxidants, herb extracts, as well as bioactive protein or peptides. This section will focus on our recent progress in the development of nutraceutical delivery systems based on barley proteins, of which hordein and glutenin are the two major fractions. So the first concept is barley protein microparticles. The emulsifying stabilization process has been applied to prepare microparticles using barley protein as the wall material. This process normally involves an initial step to form emulsions in which the protein wall material acts as a stabilizer for the core lipid. For the second step, the protein wall materials are solidified by adding a cross-linking reagent, for example, glutaraldehyde, transglutaridine, or by coacervating with oppositely charged polymers. The stabilized microparticles can then be converted into free-flowing powders using a spray drying technique. The barley protein microcapsules may be suitable for liquid or semi-liquid food applications, because the stability of the barley protein microparticles was then tested into food products, including a fat-free milk and a yogurt. about the release properties of barley protein microparticles were investigated in the simulated gastrointestinal tract using beta-carotene as a model nutraceutical. In the simulated gastric fluid with pepsin, beta-carotene was slowly released from barley protein microparticles, and less than 5% beta-carotene was detected in the release medium after 2 hours of the test. So here is the picture of microparticles. The next, okay, the next concept is about barley protein nanoparticles. So the nano encapsulation of nutraceuticals is emerging as a promising approach for delivering health-promoting substances to wide populations without harming the sensory quality of food, while providing benefits of protection and improve bioavailability. The nanoparticles can dramatically prolong the residence time of bioactive formulations by decreasing the influence of intestinal clearance mechanisms and by increasing the surface area to interact with the biological support. In addition, some of them are small enough to cross the epithelial lining of the gut and be readily taken up by the cells, allowing efficient delivery of active compounds to target sites in the body. More recently, the new technology developed for barley protein microparticles has been adapted to prepare barley protein nanoparticles of 90 till 200 nanomicron. And then the barley protein nanoparticles were biocompatible and had low toxicity, even at a high concentrations. In addition, they can be effectively internalized and accumulated in the CSO cell cytoplasms. Thus, these barley protein nanoparticles have strong potential to be used as systems of bioactive compounds to improve their bioavailability. Also, the study provides meaningful justification for further in vivo studies to evaluate the safety and efficiency of barley protein nanoparticles as a delivery system. So the next is about the future prospect. So about the future prospect, the cereal grains represent a vital and dynamic commodity that plays an important role in nourishing an ever-increasing world population. They are the most important staple food in most countries. Intense competition among food companies to address ever-evolving and sophisticated consumer expectations is driving the development of health-promoting cereal foods and ingredients. Because of their unique nutritional nature, whole grain-based food products and foods fortified with phytochemicals are likely to continue to be the subject of much research attention. These new foods will not only improve the nutrition profile of the diet, but will also potentially provide benefits such as the prevention and treatment of some diseases. Several practical considerations must further be considered before new ingredients are added into trial. Will the ingredients affect taste, aroma, texture, or appearance of original foods? Will they degrade, oxidize, or react with other ingredients and finally limit or lose their bioactivity after cooking? And then finally, since the food industry is rigorously and strictly regulated, and try and in vivo studies to assess the bioavailability and toxicology of bioactive components, including their registered products and the regulation of the intake of these functional components will be required. So, uh, uh, this is the presentation from Group 6. Maybe thank you for your understanding and your time. So maybe we just come back to Mr. Prof. Suherman.